Okay, so in this last section from this chapter where we talk about transport of messages and molecules across cell membranes, we're going to be focusing on ion channels and how they're important in nerve cell signaling. So, if we were to look at a typical nerve cell, the fundamental task or the main task of a nerve cell is to receive signals as well as to combine them and then transmit them further to their target cells or organs. So they carry these signals from one place, usually from a sense organ of some sort. For example, your eyes, your ears, your skin cells, right, to the central nervous system, where that signal is going to get integrated together with all the different types of signals that are coming through and then get passed down through the neuronal system, through our main nervous system, to the target organs, to the final place, right, where the response will occur. So every neuron has the same basic structure of the cell body, right? Just like any other cell, that's going to contain the nucleus, it's going to contain all the organelles that are needed for it to perform its everyday tasks. But then it's different than other cells in that it will have many long, thin extensions radiating from it. Usually it contains one long extension that is called the axons, that takes the signal away from the main neuronal cell body to cells far away, to target cells that are far away. And then there are many shorter extensions all around the cell body itself. And these are going to be, branching extensions. Usually these are called dendrites. And the purpose of these is, one, to increase the surface area of the cell, and also to make contact with many axons all around it in order to receive signals. So these dendrites are going to be where the signal is received into the cell body itself, and then the axon is where the signal is getting transmitted to the target cells. The axons will usually end with similar kind of multiple branching short extensions so that the same signal can be transmitted to multiple target cells at the same time, rather than each axon only sending signal to one terminal at the end of the day, right? So this kind of branching allows the cells to receive as many as 100,000 inputs from a single neuron, right? So just one neuron can receive 100,000 signals at a time, and that's amazing property for these cells. Now, there are many different types of neurons that you can see. You have sensory neurons that are going to receive signals from sense cells. You have interneurons that serve as intermediaries as the signals are getting transmitted, and then motor neurons that are going to perform some kind of final function in response to the signal that they are getting from the other types of neurons that are present there. Now, how they work is through action potentials that are mediated specifically by voltage-gated cation channels. So when we think cation, remember we're thinking about positive ions, so it would be your sodium ions, potassium ions, ions like that. And action potentials are specifically created through these voltage-gated sodium channels that can be in different conformations. Sometimes they can be either closed or open, which is obviously something that we would already think about, but then sometimes they can be in what we call inactivated state. And when they are in the inactivated state at that time, they cannot be activated at that time to restart the action potential anymore. So when a membrane is at rest in a typical condition, the channels are closed. When the membrane is depolarized, in order to, when they receive an action potential and when they get activated, they are going to be open. So they're going to allow for influx of these sodium ions into the cell, And then there's going to be a refractory period in between where the channel can no longer be activated again until it recovers from that action potential. So it's in this refractory period where it's inactivated. Once the membrane recovers its typical kind of repolarization state, the way it is typically found, it will go back to the normal closed conformation and the cycle will start again. So because the whole idea behind the way it gets activated is through that change in membrane potential, the membrane depolarization. So the inside is now, positive and the outside is negative. That's why it takes a while for it to get activated again, because until the extracellular membrane goes back to its resting membrane potential, it won't be able to restart the cycle again. If we look at these a little bit more in detail, if we were to just give it that initial stimulus, right, without having the influx of sodium ions going into it to, kind of enhance the signal, your signal is going to be very muted. It's not going to be very strong. So you need that amplification of signal through the depolarization of membrane for the signal to be transmitted appropriately and for it to get the effect that it needs. So the green line on this first graph shows that if you just received the initial signal and there was no membrane depolarization, no action potential that was moving through these neurons, you would end up with a slight depolarization, and that would be it. And that would not give you the response that you need. Now in this particular graph, the plasma membrane potential typically is set to negative 60 millivolts. So when the action potential happens, so a stimulus is put on there, right? There's a stimulus that acts upon the membrane that is going to cause the membrane, the sodium channels to open and the influx of sodium ions to go into the cytosol. There's going to be an influx, the change of membrane potential rapidly. Once it reaches that threshold potential, it's gonna go rapidly into the positive range. It goes through the 0 to positive 40 millivolts. That's the maximum that it will reach before it goes back down to the resting membrane potential equally fast. So over a course of 2 milliseconds, you had the stimulus activate this, send this signal, cause the membrane depolarization, and then go back to its resting state. So in here, if you were to look at it, it would look at this bottom graph. It shows you when you would find these sodium channels open or closed at any given time. So initially they're going to be closed at the time of the stimulus. They will open to allow for movement of ions. Once it reaches the maximum membrane potential, it will become inactivated, and it will remain inactivated until it goes back to its resting membrane potential, at which point it goes back to its closed state and can now be activated again. Remember, sodium ions, many times these sodium channels will work in conjunction with potassium ions, or channels for potassium as well. So this is what it is going to show you in this case. They are both important in proper membrane potential getting, membrane getting depolarized in a neuron for proper membrane action potential to occur. So you will have, at the time of excitation, when the stimulus is applied, you will have the sodium channels open to begin with, and sodium will begin to enter the cells. From these open channels, the potassium channels will open as it goes past the threshold potential, and potassium is going to begin to leave the cell. Now, this potassium leaving the cell is not going to make as big of an impact until basically the sodium channels become refractory and no more sodium can enter the cell. At that point, the sodium ions will continue to leave the cell, so that now your membrane potential is going to start to come back to the resting state, right? Because positive ions are continuing to leave and no more sodium ions are going back in to take that weight off. So now the membrane will slowly return to the resting state. At the point when the membrane threshold goes back to the general resting state, your sodium channels will go back to reset to their normal closed state, and the potassium channels will close. Many times you actually get extra potassium going into the cell. That extra potassium slowly, or rather more extra potassium going out of the cells, and that extra potassium slowly diffuses away so that you can get back to the normal plasma membrane potential within a very short period of time, in this case, as you can see. This is also shown here in a little bit more detail, where you can now look at your sodium channels and how action potentials are getting propagated over the membrane as the signal passes through the membrane. So remember, your neuron, if we were to draw it, you have the cell body, right? I'm not going to draw the dendrites right now. I'm just drawing the long axon because this is where the action potential is going to be enhancing, right? That's where it is getting amplified. As it is traveling through the space, right, there is a lot more area for it to cover. There are going to be a lot of these channels all along this membrane as you're going towards the terminal end, and for each channel, as it becomes activated, it's going to start pouring in these the sodium ions. So the membrane potential is going to get amplified, and the membrane is going to get depolarized as you propagate the signal down the axon. So here you have a closed channel. When it gets activated, it's going to become depolarized. It's going to open where the sodium ions are going in. That is going to then activate, depolarize that neuron and activate that neuron, and the signal will continue to move down until it reaches its final destination. So you go in this cycle, right, in this case, where they are closed, they are inactivated, they get depolarized, they open, sodium ions are going in, and then they are going to get closed again once they have gone back to their normal state. One thing you want to be aware of is that action potentials can only travel forward from the site of depolarization. So even though these are closed and these are technically, could be activated, they won't be, because the site of activation is right here where this blue area is, where the depolarization is. So the only channels that are going to get activated and that are going to amplify the signal are going to be at the site of depolarization forward. Those are the ones that are going to produce the action potential, and then it's going to propagate from there, moving forward to the terminal end until it reaches the terminal end. So, that's how your action potential is moving along the length of an axon. Now, what happens when you get to the very end, right? So, let's say, now you have done, you've moved your action potential all along, and you're ready to move on to the terminal end where it's going to now transmit the signal. Well, once it reaches its target cells, it's going to connect to the target cells at the synapses, right? So there is going to be a very particular type of connection there. That's going to happen through, again, smaller extensions that are going to happen. They come out of axons, and that's going to be your presynaptic nerve terminal, and it's going to meet the cell that it's going to activate at the dendritic postsynaptic nerve cell, right? So this is going to be your postsynaptic nerve cells membrane, and the one over here and here are your presynaptic nerve terminals. Now you are going to see an influx of ions accumulating at this point, and these ions are actually calcium ions. So voltage gated calcium channels in these terminals convert this signal from this electric output, this electric signal in the shape of ions, into an actual chemical signal in the form of neurotransmitters that can then move into the postsynaptic membrane and activate it or deactivate it. So the electrical signal that is passing through the axon is going to get converted into a chemical signal at this point. So when it's released from that synaptic vesicle at at the edge into the synaptic cleft and reach the postsynaptic cell, it's going to be a neurotransmitter of some sort, like GABA, right? That's a neurotransmitter that can be bound to the actual postsynaptic neuron receptor, a neurotransmitter receptor, and activate that cell's responses in response to it. These are done, this happens through voltage gated calcium channels. So you have an influx of these calcium ions that are going to go into the cell, that are going to move through the membrane at that terminus, and they are going to activate the release of the synaptic vesicle. As the calcium ion goes in, when there is an activated nerve terminal, the voltage gated calcium channels open up, calcium goes in. As it goes in, the vesicles containing those neurotransmitters will fuse to the plasma membrane and release their contents into that small space in between the two cells called the synaptic cleft. And that synaptic cleft is where it will find the receptor of the for that neurotransmitter that can bind to it and then activate the postsynaptic cell in response. Okay, so once again, once this activator, once this neurotransmitter has bound to the receptor, it's activated that receptor, it is going to then send the signal through change in membrane potential. Again, it's going to get converted back into an electrical signal in this postsynaptic membrane, and as a result, ions will get influxed into the cell to activate the appropriate response. So the activated postsynaptic cell is going to look very similar in some ways to how we just looked at the axon transmitting that action potential, right? The neurotransmitter is not getting into the cell, it's just getting bound to its receptor on the membrane, which causes it to get activated and open up, which causes an influx of ions inside, changing the membrane potential again, causing an electrical signal that is going to be essentially leading to the response that we are going to be looking at the end of the day. Sometimes you will see that, or many times, there's going to be excess neurotransmitter that was released that has no place to bind, because everything, all the receptors on this postsynaptic cell membrane that are in that cleft have been bound already. That excess neurotransmitter usually gets removed, reuptaked, or reused by that presynaptic cell for either enzymatic degradation of the neurotransmitter or for reuse, depending upon what it is that it is going to do next, whether it is going to get reactivated or not. An example of these is an acetylcholine receptor at the neuromuscular junction. They are ligand-gated, so they only get activated and they only open up when acetylcholine is released from the presynaptic cell and gets bound to the acetylcholine receptor at that neuromuscular junction. So there'll be a nerve cell on this side, obviously, and it's going to activate the muscle cell on the final end. So the acetylcholine binding sites are there. They are always hanging out, but they're not going to be activated until acetylcholine binds to it. Two acetylcholine molecules binding to it activates this receptor to its open conformation, causing sodium ions to get moved into the cell membrane, into the cytosol. And when they move into the cytosol, that's going to obviously change the action, the membrane potential, as we've talked about. So membrane potential gets changed, and that will cause the muscle to do work as whatever work it needs to do, whether it's contract or relax or do get excited and moved. So ion channels are essential for proper nerve cell signaling, and if they weren't working properly, you wouldn't be able to transmit the signals throughout the body and do the work that they need to do. So neurotransmitters are chemical messengers that are used to transmit the message from the nerve cell across the synapse to the target cell. However, the signal initially that is getting transmitted through the axon is through ions, through influx of ions into the cell, into the cytosolic space that causes the membrane depolarization. That leads to an action potential getting built up and transmitted all along the axon until it reaches the final end, the terminal, the neuron terminus, where neurotransmitters are going to get released. These neurotransmitters can be either excitatory or inhibitory. So they don't always have to cause muscle contraction. They could be muscle relaxants. It depends on what type of signal was received by the cells at the end, right? The complexity of synaptic signaling enables us obviously to do all the work that we do, to think the way we act, to respond to the stimuli around us, to hear, learn, remember everything. And many psychoactive drugs will affect the synaptic signaling by binding to neurotransmitter receptors directly, inhibiting their function and not allowing them to function properly. Pain receptors, right, can be, some of the pain receptors can be made useless or can be inhibited so that you don't receive as much pain by using drugs that bind to drug molecules like Tylenol that bind to these receptors and inhibit their proper functioning, inhibit them from transmitting the signal the way they normally do so that you don't feel as much pain. So neurons will receive input from thousands of other neurons, just like we talked about. A single neuron can receive up to 100,000 different signals at a time. And for each one of these, you're going to have these presynaptic terminals that are interacting with the cell body and the dendrites of the a final neuron, and they can, any one of these can cause an action potential to occur and can cause an effect to be seen. So now different types of neurons will get activated differently, right? So one of those is light-gated ion channels. These are specific types of channels that can be used to activate or inactivate neurons in living animals because they are controlled by light, and these are very easily controlled because it's not technically non-invasive, right? You're just switching the light to activate them or to suppress them. So here's an experiment that was done by Lin et al. That shows how these channels work. So in this case, a gene encoding channel rhodopsin was introduced into the hypothalamus of, or some of the neurons in the hypothalamus of the mouse brain. Now, this particular gene is going to get activated when you activate blue light, when you introduce blue light to it. And they did that by actually, it is a little bit invasive, I guess, because they are actually using a fiber optic cable that's implanted into the animal's brain to expose it to blue light. When the blue light is there, when you essentially switch on the blue light, it opens the channels in that area that were, encoding this channel rhodopsin. Around that, those neurons get activated, they depolarize and express it, causing the mouse to become extremely aggressive. However, if the light is switched off, they become go back to their normal behavior and become docile again. So when the channel is open, the sodium ions are going into the cytosol, depolarizing the plasma membrane and causing that aggression response. So this is how effective these channels are. They are very quick to respond in response to whatever environmental stimuli they are meant for, and cause an action potential to cause a response. Plants also communicate through ion-gated channels and secretion of signals, both when they are just communicating with cells within that plant, but also to the environment itself. And we can look at that also by looking at how various parts of the plant are generating similar kind of action potentials and similar membrane potential changes, membrane depolarizations in response to certain environmental stimuli. So here in this particular plant, this is one of those carnivore plants that will eat bugs, right? The presence of the bug, just a small aphid, is enough, activates the membrane potential in those cells that it touches enough for it to snap shut. It is an extremely rapid response where the depolarization happens in a very quick fashion, causing the leaves to shut, and thus, obviously the aphid getting stuck inside, so it can be consumed by that plant. So again, these Venus flytrap are a classic example of how quickly these signals can pass and affect, and cause a response at the end. And if you look at this, it's less than half a second for each of these activations to occur. So it is very quick response at the end of the day. Okay. So we are gonna look at answer any questions in class on Tuesday. Until then, you can text me your questions by email. Take care.